Good evening. My name is Jude Fitzgibbon. I'm a research scientist at the Barts Cancer Institute in London, that's part of Queen Mary University of London. It's really a great pleasure to present to you today a summary and showcase the work of the lymphoma science subgroup. The group itself is hardly 12 months old, and it's chaired by both myself and Dr. Cathy Burton from the University of Leeds. And I think the plan really is to develop and support lymphoma research and researchers throughout the UK. And we've brought together a multidisciplinary group of researchers to complete this task from 14 centres, and they include basic, translational, and clinical scientists. And here they are, all from the University of Glasgow, and Professor Ruth Jarrett, a pathologist, down to the University of Sussex, and Dr. Simon Mitchell, a computational biologist, and many researchers in between, from trialists to basic researchers. I think the overarching aims of the group, and really this was the brainchild, I think, initially of Andy Pettitt and Kim Linton, was to really create a UK-wide research network comprising all types of researchers to try and increase innovation, competitiveness, and build on the existing successes that we have in the UK. Also an opportunity to engage funders, patients, and pharma, and to ensure that translation research is part of the conversation of every new clinical trial. That's a key point. Support early and mid-range lymphoma researchers, and offer additional peer review and mentorship, which I think is very good at the individual centres, but can always be improved, and to increase the innovation and competitiveness in order to secure funding. Now, we need to build on the existing infrastructure and connecting, I think, UK lymphoma researchers together. And we see the successes are very much based on that connectivity across the UK, with success stories like PMEL, the strength of a large cadre of early career researchers. We also need to maximize the potential of our existing tissue collections from several different sources throughout the UK. I think we all know Peter Johnson. Peter leads the Precision Medicine and Aggressive Lymphoma Group. It's an initiative that was funded by Blood Cancer UK and comprised 5 different centres throughout the UK working together on precision medicine and aggressive lymphoma, typically diffuse large B-cell lymphoma. We've collected over 5,000 biopsy samples as part of PMEL at the present time. And really, I think this work fed off the success of a clinical trial, the Remodel B trial, and the use in real time of gene expression profiling studies, translational studies, to actually define patients that were suitable for randomization within the trial itself. I think this type of approach is being followed in relationship to primary central nervous system lymphoma. Again, both retrospectively and prospective collections from 3 different trials of both samples, and providing an imaging repository and clinical data, with the opportunity therefore to go forward in comprehensive molecular profiling and following kind of dynamic biomarkers over time. I think the key point here is again, the work across 3 different institutions, the University of Nottingham, University College London, and Queen Mary University of London, with Chris Fox, Kate Konarski, Steffi Trust, and Jessica Ockerson. I'm very impressed with the quality of the early and mid-range researchers that we have here in the UK. And here are 6 that are on the lymphoma subgroup panel, many of whom have actually gained experience abroad in the US. Dan Hodson worked with Lou Stout in diffuse large B-cell lymphoma in Bethesda at the NIH. Sean Lim worked with Ron Levy at Stanford. And we've also got a kind of expertise across several different major lymphoma subgroups. Chris Carey in relationship to Hodgkin's disease, Simon, his work on pediatric Burkitt's lymphoma, Dan Hodson, diffuse large B-cell lymphoma, Jessica Flickler lymphoma in particular, but also now central nervous system lymphoma, Sean Lim running trials and basic research in immunology, and Simon Mitchell, a computational biologist, funded through Cancer Research UK, the MRC, and several different funding organizations. I think we also have to keep in mind the importance of collecting allied both clinical and primary material from patients to have these as a repository for translational research and to understand the natural history of these diseases in the cohorts in the UK. In relationship to the T-cell lymphoma tissue bank, Matt Ahern leads this in Nottingham with the collection stored and collected in Liverpool through Andy Pettitt and Melanie Oates. You know, and a large number of sites have contributed. Probably people listening to the talk here today have contributed biopsy samples, 32 UK sites, over 100 patients, and a number of sample types. I think the same is true in relationship to mantle cell lymphoma that's now run by David Lewis in the University Hospital Plymouth. Again, a large number of patients and primary biopsy material have been collected from these patients over a period of time, allowing for allied clinical and molecular and indeed other analysis to be performed. 600 patients in total across 68 sites. And I think both these tissue banks provide a framework and an opportunity to develop translational programs, basic discovery programs, and to compete with similar programs that are ongoing internationally at the present time. I think in conclusion, you know, I think translational research and the individuals we have are very strong, but the question really is it greater than the sum of the parts? You know, we need to develop this multidisciplinary team and approach because it's worked very, very effectively in relationship to PMAL and other initiatives. And I think what we want to try and do is to develop a program whereby not only is translational research taught about in relationship to clinical trials and clinical and collecting primary biopsy material, but we also need to feed and to encourage the next generation of basic researchers to be engaged in lymphoma research. And the opportunity, therefore, for not only discovery science, but actually to introduce new ideas, new potential mechanisms, new potential types of biomarkers and technology, whether it's engineering and nanotechnology, whether it's the value of model organisms, new biochemistry, new computational approaches, whereby going forward, in order to be competitive, I think internationally, we have to think outside the box and not just basic gene mutation and gene expression and the analysis of a set of samples, but to actually incorporate the value and the strengths that we have in basic research here in the UK. So I think in conclusion, you know, I think the science subgroup has really pulled together a fantastic cadre of individuals with a wide range of experience who hopefully can support and build on the reputation of the UK in clinical and translational research. It'll offer a forum to support lymphoma and allied researchers in the UK. And we need to try and ensure that translational research is part of the conversation of every new clinical trial that's being opened and considered in the UK. This is the website where you can get information on all the different members of the science subgroup, and they can all be readily contacted. We're minded by Francesca Paradi, who's the research group's coordinator, and both Kathy and myself will be very happy to entertain any correspondence from those interested in the subgroup and how we might actually help the community going forward. As I said, thank you all very much for this opportunity. The very best.